So today I'd like to tell you about my project on using long-read capture sequencing to identify novel RNA isoforms of psychiatric risk genes. So alternative splicing is a key regulator of neuronal differentiation, and it happens in 95% of human protein coding genes. And aberrant splicing is associated with both neurodevelopmental disorders, such as schizophrenia and autism spectrum disorder, but also neurodegenerative disorders such as dementia and ALS. To better understand the mechanisms of these disorders, we can try and annotate the splicing associated with them. Pathways linked to complex psychiatric disorders and neuronal development are often enriched for these long, complex genes with many splicing isoforms. While average human protein coding genes encode only 3 to 4 RNA isoforms, which are 1,000 to 2,000 base pairs long, some giants, such as the neural cell surface molecule genes, encode hundreds of isoforms that are functionally diverse. The size and complexity of such genes make accurate transcript identification and quantification by standard short-read sequencing methods extremely difficult. It is also difficult to identify how all of these isoforms contribute to the functional protein diversity. But in cases where a gene's differential protein isoform has been described, such as the case for neurexin genes, it has been shown that differential exon inclusion can have an effect on cell type expression and subcellular localization. One example of such a long and complex gene is CACNA1C. CACNA1C is associated with many psychiatric disorders, and according to the current annotation, has 56 exons and 31 isoforms. Studying genes like CACNA1C using short-read sequencing methods is difficult. Therefore, we use long reads to capture full-length isoforms of such genes. The other issue with genes like CACNA1C is that despite being fundamental for brain function, it is lowly expressed in the brain when compared to other genes, and CACNA1C's own expression in other tissues. Annotating tissue-specific low-abundance isoforms of genes like CACNA1C using unbiased transcriptome-wide sequencing method would just not be cost-efficient. Therefore, our collaborator Nicola Hall of Tunbridge Lab used an approach that combines probe capture enrichment and long-read cDNA sequencing to profile transcripts of 1,469 target genes. So these genes included both protein coding, psychiatric risk genes, and also long non-coding RNA genes with GUS hits. Because long non-coding RNA genes are less well annotated, accurate probe design was significantly more difficult. And then to control for the quantification biases that were introduced by the capture enrichment, Nicola used synthetic spikins at known concentrations that were added to the cDNA mix. Capture sequencing was run on 52 Lieber Institute post-mortem human brain samples from patients with psychiatric disorders, but also from both fetal and adult controls. Prefrontal cortex, hippocampus, and caudate were selected for the study because they're known to both be transcriptionally and functionally diverse and are also known to be strongly implicated in psychiatric disorders. So first we checked if enrichment worked, and gene-level quantification has shown a nearly 50-fold enrichment for target protein coding genes as compared to genes not in the capture design. As you can see on the left, these enrichment factors match the ones that were observed using qPCR controls. As you can see, targeted spike in transcript abundance was strongly enriched with the input concentrations. Unfortunately, long non-coding RNA genes were not significantly enriched, but I'll address this in the next slide. So to check if lower post-capture enrichment of long non-coding RNA genes was not biased by the lower number of probes designed for these genes. We plotted them, their measured TPMs against the number of probes, and as you can see here, capture enrichment was not biased by the number of biotinylated probes that we used to pull down the isoforms. Since the preliminary results suggested that the dataset can be used for quantitative expression comparison between samples, I then needed to create an annotation pipeline to create a custom annotation for the 1400 target genes. This annotation pipeline involved restrander and 2-pass tools that were used for correcting the long reads. And then I also used isoquant and DAMA for isoform detection. So thanks to the work by Nicola, I had a ground truth I could use to validate each step of my pipeline. So my ground truth was this alternative first exon of CACNA1C that was previously confirmed by PCR. So even though our REN values were high, there was still some RNA degradation. And then this alternative first exon is the sort of isoform complexity that is really hard to identify, as the 5' splice junction is also a transcript start site, and therefore was highly variable, as you can see here, between the reads. Some tools therefore identify this variability as an error, an erroneous shift in the terminal exon starts, and therefore it removes the exon from the final annotation. It is difficult to define transcript start sites without additional experimental steps such as 5' race, which is why it was important for me to select the right tool for this step of the pipeline. Using TAMA and isoquant and our updated transcriptome annotation to quantify isoform expression, it led to principal components explaining more of the inter-sample variance and resulted in clearer sample separation, as you can see here. And that was especially true for the fetal samples. And as you can see in this table here, some of the novel isoforms that I identified using my pipeline were involved in significant isoform switching events between samples. Isoform switching events mean that there was a significant change in the relative contribution of an isoform to the overall gene expression between conditions. There were 95 isoform switches between adult and fetal prefrontal cortex samples, and 79 of those involved novel isoforms. An example of such a switch can be seen here in CACNA1F, a calcium channel gene. As you can see, there's, while the annotated long channel encoding isoform makes up a quarter of the adult expression and contributes the most to the expression of the gene, the truncated isoform makes up 80% of the fetal gene expression. And finally, you can see that we could use long-read Nanopore capture sequencing to profile transcripts at a sequencing depth that was just not affordable before. We identified over 56,000 novel isoforms for 1,100 genes of interest, and we could reliably quantify their differential expression and usage between brain regions, both in control donors, but also between disorder cases and control samples, which was validated through independent data sets. I finally would like to thank everyone in my lab, including Wilfred Herty, Ian Macaulay, and David Wright. I would also like to thank the members of the Tunbridge Lab, Nicola Hall and Paul Harrison. And I would also like to thank all of the anonymous tissue donors.